In this interview, I talked to Dr. Dickson Despommier. Don't worry, he's not as French as his name sounds. He has, however, had a pretty full life. At 82 years old, he's quite spry and like sharp as a tack. He's also had an incredible life, as if he had multiple lifetimes all smushed into one. He was a parasitologist at Columbia University for 27 years. Then he was an ecologist and gave a number of talks all around the world about vertical farming. The South Korean government even erected a vertical farm and credited him for inspiring them. He's a professor emeritus. I don't know if you know what that is, but it's Latin for fucking amazing professor at Columbia University as well. So he's like better than most Columbia University professors. He also has, just casually, two podcasts with a total of 6 million downloads a year. This Week in Virology and This Week in Parasitology. And if that isn't enough, he also has some really beautiful art. Depommierphotoart.com. I'll put the link in the description. So this episode was quite contentious. We had some disagreements about gain of function research that I don't think were fully resolved. But contention is part of life. If you don't disagree with people, life would be super boring if we all got along. That's for sure. So I would love to hear your comments about what you think of this video and what you would, if we do another interview, what you would like to hear from him. And hope you enjoy it.
So what made you choose me?
Okay, good question. So
I'll interview you for a minute.
Yeah, sure. So I was looking at, there was this bounty online. I'm part of this like bounty group where they pay people to do various things. And I saw this woman and she said, I'm against gain of function research, and I will pay a year's salary for anyone who can write to all the top universities and tell them why they shouldn't do gain of function research. I want to go out and talk to all the students who are going to go to that university and tell them about it, tell them why they shouldn't go to that university. We need to cut them off. And I said to myself, I want to talk to somebody who likes gain of function research and ask them about what they like about it. And so I found Scientists for Science, and I looked through a lot of different scientists, and I was looking specifically for people with interesting projects that are interesting to me, and also who can speak and have something to say that isn't, sometimes, like one time I talked to this scientist, not on my podcast, but I was talking to him about like water bears, those little tiny,
tardigrades, little tardigrades. I'm very familiar with tardigrades.
I'm sure you are.
They'll survive on Mars.
It's crazy, and in space, oh my God.
Nothing hurts them.
almost absolute freezing too. It's crazy.
Have you ever read The Hitchhiker's Guide to the Galaxies?
Yes.
They shouldn't have used dolphins. They should have used tardigrades.
Yeah. But
when they left the Earth, tardigrades would say, "So long and thanks for nothing." It's not about fish. It's about, we don't need anything. We came, we looked it over, and we said, "Nope, not for us. We're getting out of here." And they leave nothing. There's no way to know if a tardigrade has actually been somewhere.
Really?
Yeah,
they don't consume?
Leaves no trace.
Huh.
Yeah. So how do they live? I think you should interview a tardigrade expert and say,
I think I will.
This probably didn't come from this planet.
Yeah. You think so?
It's possible. It rode in on a comet. Who knows?
So,
but we don't want to go in that direction with this conversation. I think I want to talk about gain of function, because if I were a student listening to this woman's invective about something that I don't know anything about, the first thing I would want to do is to participate in it, to find out what it is.
Yeah.
So that would increase the enrollment at that school. It would not decrease the enrollment in that school.
Yeah.
You get more students, because youth is so curious and so limitless in terms of boundaries of thinking, that the best way to get something done is to teach. And we'll get to that, because the idea that I guess I've become known for is vertical farming. And that's later on down the road on your interview, but it arose out of a need to fill a gap of my own intellectual growth. But first, let's talk about virology, because since you're part of this podcast, what is the name of your podcast?
I don't have a name yet. I
You
I
don't
don't
have a name?
I don't know if I have a specific podcast as such.
Well,
I have a
I bet you
channel.
do.
It's called Serene Desiree. It's my name, and then I
I
go on and
understood. It's a great name, by the way. I meant to
thank
tell you, it's
you.
a fabulous name.
I like this Dickson Despommier too. It's got the alliteration and it's very French.
It
Um,
means apple trees.
oh,
Last name is Despommier, of the
Despommier.
apples, of the
Oh,
apples. But
huh.
I'm not sure it's French. It might be Belgian also.
Well, they
So
spoke
we're
French.
not really sure about our origins, but it's close enough.
And
So,
you were born in New Orleans, as
bon non,
New
ça
Orleans,
sure
New
was.
Orleans. So is that, do you have then they're a very French town, a very French influenced town. So
Yeah, it's French, all right, but it's kind of funny French because the Michelin Guide in France doesn't recognize any of the restaurants in New Orleans, in Louisiana, in fact. They call them faux French
huh.
because they don't really follow any of the rules. Why would they? They make them up, and
Yeah.
they have their own cuisines, and everybody should be allowed to have their own cuisine, I think, except tardigrades because we know they don't eat anything.
They don't. Yeah.
So, yeah, so New Orleans has a bad rep in France, but it has a wonderful reputation throughout the
Everywhere
rest of
else.
them. Yes,
Everywhere?
it even has 3 Michelin star chefs working in New Orleans, and their restaurants are shunned by the Michelin people. I don't get it. I really don't get it.
Do you know what the
There's,
origins of Michelin, of the Michelin stars?
yeah, the Michelin started in France, some way to standardize French cuisine so that you could judge it.
Well, yes, except before that. So you know the Michelin Man, you know the
Oh,
tires.
you know this? You know this?
Yeah.
Okay, fine. Go ahead.
The tire makers wanted people
Oh,
to
yeah.
drive more because they were the first tire, I don't know, the first tire makers, but they were like early on, and they
Yeah,
wanted
sure.
people to take their cars and go around the country. And so the Michelin tire people, the rubber people, started rating
How
different
funny.
restaurants, and so people could go and drive around the country
Oh, that's very
with their
funny.
tires. Yeah.
I did not. I might have known that, but I've since forgotten, unfortunately. So following up on that, of course, the Michelin people would instantly approve of escargot for 2 reasons. One is it's a pun on 2 snails looking at a car, and one says to the other, "Look at that escargot." It was an electric car, of course, and the other one is, of course, that they're interested in rubber, and that's exactly the way escargot tastes. If you eat escargot, it's about garlic, parsley, butter, and rubber.
Wow. Never had it. I don't know if I ever
So,
will.
no, well, most people say, I don't eat snails. Well, you should try them. They don't actually have a taste. They're tasty because of the flavorings. So
Ah.
Michelin has grown as a kind of a snobby little group of people that sit around a table and decide whose cuisine they should respect the most. And believe me, the chefs that get Michelin stars make huge amounts of money. So it's about money as well. And I once participated in the cuisine. There's an international cuisine conference held I believe it's every 4 years in Lyon, which is the epicenter for cuisine in France. And I actually give a talk on vertical farming, which they liked because it supplies them with fresh produce, obviously. So for me, that's a gain of function.
Why is that a gain of function?
Well, because they didn't have fresh produce before, they had to wait for the harvests. Now you can have it anytime you want. So that really does deconstruct all of the famous seasonal restaurants, because there are no seasons indoors. So you can basically have whatever you want year round. And so therefore, chefs that say, well, I only cook seasonal food because I support my local farmers, your local farmer is going to be growing it in that building over there. So you better watch out. Better change your attitude.
If it's okay with you, I think the podcast has started. If that's fine.
No. I want it to start. This is perfect. Perfect. We'll get to gain of function. Don't worry. We'll get to gain of function.
Wonderful. Oh, my camera has just turned off. That's a good time to announce that we're
Whoops.
starting.
But the C922 has never ever given me a problem.
Yeah, it looks good too.
And it's a high definition. I'm on several podcasts, and so the guy who runs them insists on the finest high-tech equipment. And this thing is not expensive. I think it's a couple $100, but
Wow. What podcast are you on?
I'm on two that I established. The first one was called This Week in Virology, and that started back in 2006, I think. And believe me, we have covered the subject of gain of function ad nauseum. And so I'm not a virologist, but I got roped into it because I was considered a good teacher. So the guy who roped me into it, and I didn't realize what I was getting involved with. It's been ongoing now. We've never missed a Friday.
Wow.
Since 2006. So we're up to episode number 865, I think.
Oh my God.
So we're approaching 1,000 consecutive webcasts, or podcasts, I should say. The other one I'm on is called This Week in Parasitism, and it's about my subject because I'm a parasitologist.
Wow. That's,
and then I used to have one which is still running. You can still access it. Your camera came back on. That's good. Great. The other one, the third one, is called Urban Agriculture. And it's interviewed about 35 vertical farm CEOs and their personnel. And then they stopped giving me permission. They didn't want to come on my show. And the reason was they thought they were in competition with the other vertical farmers. So that didn't turn out to be true. Yeah, but that's the way it was. So there are several podcasts now on vertical farming, but I don't run any of them. I have bigger fish to fry.
They have to stop talking to each other because of competition.
Exactly. They didn't want them to know what they were doing. Mm-hmm. And it's so sad because the more people that know what you're doing, the more the field will grow, and then the more robust it will be, and then the less vulnerable it will be to shutting them down. Mm-hmm. And they don't actually see it that way, but they have started to see it that way, by the way. Mm-hmm. So I might revive it, but it's a lot of work, as you know. Mm-hmm. If you have a regular schedule, and the two podcasts that I'm on, the first one, This Week in Virology, they actually mean This Week in Virology. Yeah. The other one is This Week in Parasitism. Oh, hello.
That's wonderful.
How you doing?
He fixed everything.
Yeah.
Excellent.
Dickson. Yeah, cool. Thank you very much. Mm-hmm. I have already missed weeks. Actually, I don't have a fixed schedule because I I should and it's better for the algorithm, but I think my biggest weakness right now is organizational skills. I have to be honest with you, as you might have noticed.
Join the group.
Yeah. But I just decided I just need to keep doing it, and then things will fall into place. I will get organized as I go, and I can't originally I had this idea of I'm gonna keep a fixed schedule, but actually the looming nature of a fixed schedule over me made me produce less somehow.
I don't blame. No, of course. Yeah. That's natural. But the best way to overcome that, we learned really soon after we decided to do it, was to get a partner and to podcast together. And so you both sort of feed off each other's energies, and you can maintain the flow of the information better. You're not all stuck out there by yourself. You're not totally responsible for anything. You share that responsibility. And I think that works well for Vincent and myself. And now we have 5 people that are steady participants in our webcast. You should go to it sometime. It's all free, of course.
I would love to. Yeah.
Called This Week in Virology, and you can actually look up the episodes in which gain of function was the main topic.
Okay, so gain of function. Can you tell me?
Yeah.
Let's
talk about
gain
this.
Of function research?
Sure, of course. I love this subject. So this requires giving you a little background in molecular biology, which I'm sure you're very familiar with. So I will just say that pieces of DNA produce things And the things that they produce are called proteins. And proteins do things. They're the engines of your body. All right, so the blueprint is the DNA. And the product of that is the proteins that hold you together and that make you who you are. Proteins. Well, there's stuff also like lipids and saturated fats and non-saturated fats, but they are not encoded by the DNA. The DNA encodes proteins that then make those other things. So they're machines of your body. Okay, so now that's your body. And you've got 23, probably 23, well, you've got 23 chromosomes. You've got something like, we're not exactly sure the number of genes that we have. So each gene makes a different protein. But we have, let's say we have between 25 and 30,000 different proteins that we can make from our genome. The whole thing is called the genome. It's big. It's not as big as some things, though. You'll never guess what has the biggest genome of any living thing. I'll give you a hint. It's a flower.
Oh, a flower.
And it's one of the most beautiful flowers you'll ever see. And they raise them in greenhouses. And you use them in corsages. When you go to the prom, you will require that your beau brings you a daisy? No. A tulip? No. An iris? No. An orchid?
Ooh.
An orchid?
Oh.
Orchids! Okay, so the orchid, if you look at their genome, they've got a lot of genes. They've got so many genes, they don't know what to do with them. They've doubled them up, tripled them up. So they're called ploidy. If you have one level, you got ploidy. If you have two, you've got diploidy. We're diploids. We have two copies of every gene in our body. You've got one from your mama, and you got one from your papa. And you're stuck with that. And you don't always turn on all the genes. Sometimes you turn on your mama genes. Sometimes you turn on your papa genes. Depends on what you're making. That's very simplified. It took a long time to find that out, however. Okay, so let's talk about other things that have DNA, but we don't consider them alive. What could you think of that has DNA as the basis for its existence, but it is not a life form per se?
Is it a virus?
Yes. Yes, you're very good at this. Okay, so you get A+. So what the heck is a virus? Right? So we have two kinds. We have DNA viruses and we have RNA viruses. What the heck is an RNA? Well, I will give you a hint. Well, I'll just tell you what the heck. Right. So the point is that if you don't use common language and if you don't appeal to people's empathies and sympathies at the level of, you know what, I don't know everything, then they will never forgive you the way you teach. They'll think you're preaching at them. But if you include them in the conversation, and if you add a little humor, and if you add a little humility, and you are interested in whether they understand what you've said by listening to their responses, then you become a super teacher. And very few teachers do this, and I learned this from somebody. I didn't make it up. I learned it from somebody. So I'm leading you through the wilderness of genomology to get to the issue of what is gain of function, right? Okay. So viruses contain what we would call, these pieces of DNA and RNA are referred to generically as nucleic acids. The word nucleus, though, doesn't apply to a virus because it doesn't have one. So what the heck is a nucleic acid doing in a non-nucleus organism? Wait a minute, I said organism. I didn't mean that because viruses are not organisms. How do I know? That's very easy to know that one. What is an organism? An organism is an entity that consists of nucleic acids, of course, and proteins, and that has a metabolism. Metabolism is the key. So when you breathe in and breathe out, you're breathing in oxygen and you're breathing out carbon dioxide. You're converting the glucose that you consumed in food to CO2 to get it back in the atmosphere so the plants can take it up, so they can make glucose so that you can take it up. So the sounds like a circle, right? Okay. It is a circle. It's a cycle. But viruses can't do that. Viruses have little pieces and snippets of your DNA. How the heck did they get that? They learned how to do it by mutation and random mutation. That is, it gets a little bit more complicated now. If you look at the structure of DNA, it's like an alphabet. It only has 4 letters. Imagine ATGC, ATGC. They stand for something, but don't worry about what they stand for. Just make the assumption that you can mix them all up. You can get sequences in a row that go ATTTGGGCU, and when you decode those sequences, they correspond to the amino acids that your proteins are made out of. It's a miracle. But somehow the message of the DNA has to get to the cytoplasm of the cell, because that's where the machinery for making proteins is. The blueprints are in the nucleus. Oh, okay. So the message that comes out of the nucleus out into the cytoplasm is called ribonucleic acid, or RNA. And it's a sort of a mirror image of DNA. When I say mirror image, A's line up with C's and G's line up with U's. So they coat like this, and then they make a spiral. And that spiral is what DNA actually looks like. And it's cool stuff. It has kept people busy ever since they discovered it. And it's taken a long time to come to the conclusion that what's inside your nucleus is the blueprint. And what's outside your nucleus is the result of translating that blueprint into a structure. And that's simple biology that really is complicated. Okay, that's simple biology. Every cell in your body except there are some cells that don't have nuclei, like your red blood cells. They don't need them, but almost all other cells in your body are talking from their nucleus to the cytoplasm to tell them to make something. Make me more of this, make me more of that. Why do you have to make more of something when you've already got it? And the answer is that the body doesn't just need proteins, it needs new proteins. Because the old proteins tend to break down just like machinery, just like your camera did on your machine. So when you fixed it, what do you think you did? You gave it gain of function. It was broken, you moved in, you pressed a button, boom, it came back on again. Ha, I just gained my function, yay. Well, we can't do that with proteins, because proteins, when they wear out, they degrade. And they fall back into a heap of amino acids that the body can then reuse to make other proteins, for instance. So it's a constant boilerplate of manufacturing. If you opened the door, you'd swear you were watching a car being made from scratch. You can take ores from the ground that are metals and then press them after you've melted them down and turn them into sheets that you then press into shapes that you then assemble into a car that you drive out of the car manufacturing plant, and you've got yourself a car. Well, that's what happens when the sex cells come together and make a baby. The embryogenesis leading up to the baby itself is the assembly point for making a human being. And it's very complicated. Okay, but viruses are not complicated. Viruses are very simple. Viruses are little bitty pieces of your DNA that somehow the DNA itself mutated in such a way, and this is an hypothesis now because we've never observed it. It gave rise to a number of mistakes. So viruses, we think, are natural mistakes.
Are you saying that not my DNA, maybe my DNA, but people's DNA somehow, while it was replicating, created, like maybe there was an offshoot of some DNA that actually created a virus that came out of it?
Yep.
Wow.
Well, yes and no, because that's true for some viruses, but other viruses probably arose way back when life itself arose, which is right now, if you look it up, it's about 3.53 billion years ago is where we can first detect in rocks the fossils of bacteria and algae. And so you have to go back before that in order to say that maybe it was another billion years, who knows, give or take a billion, who cares? We've only been around for 4.9 billion total. So we have some wiggle room. But some people believe that viruses arose very soon after life as we recognize it arose, to take advantage of the life that was there. It's a parasite. So viruses need your cells in order for them to replicate, to divide. They can't do it on their own, so they have no metabolism. That's why I say that they're not life forms, because all life forms can do that.
See, viruses are parasites. I want to.
They're incredibly pertinent because they steal little pieces of your DNA. When they replicate, where do they replicate? Some of them replicate in the nucleus, your nucleus, and so they have ready access to your DNA. Where do they replicate inside your nucleus? It depends on the best match that they can find for their little sequence. And so now it attaches to that sequence. And from that point on, it tells the cell, well, make more of that. That's how we make more of ourselves.
So a virus is floating inside my body, and then it finds one of my cells, and then like injects.
Somehow it gets in.
It gets into it, and then it looks through it scans my DNA. It looks through my DNA, and then finds something that matches it, and then
it mirror images it.
ATGC,
ATGC, remember? It has to find something that lines up, that hooks it up. It's like Velcro.
Okay, wait. So, for my understanding of DNA is that it's double-sided, but it
has to open up in order for you to access the genes.
Okay, and then when
it's a spiral, it's all tied up and nothing's happening.
Yeah.
The moment it opens up, now you can replicate something.
And what is the virus matches with my DNA?
It sends a signal to your cell to replicate whatever this is. Okay, so there's an enzyme called DNA polymerase, which actually makes a copy of whatever just did that, and that turns out to be the virus. Now that's some viruses; they don't all behave that way, but some viruses do. So I would highly, if you're interested in this subject, and if you want a simplified version of virology that you can really understand, that's really well done, I will give you a website to go to. It's monitored by and managed by Dr. Vincent Racaniello, my partner, that we do This Week in Virology. And if you go to the website, This Week in Virology, you will see as part of the show notes where you can go and learn more about viruses. He's got a Coursera course, and he's also got a virus course to the Columbia University students. And he offers those online to anybody that wants to take them. Whoa. And they're fascinating. They're absolutely fascinating. I mean, it really blows your mind to realize that there are things like that going on out there, right?
Yeah.
Until you hit a pandemic like this COVID-19, once that happened, everybody wanted to know about what a virus was. What the heck is this thing? It's called a coronavirus, right? They have various names for these various, you've heard of polio, you've heard of rabies, those are viruses. Herpes, those are viruses. There's thousands of viruses, perhaps even millions of different viruses. They don't all infect people, though. So some are very specific for people, and some are very generic, and they will infect all animals, like West Nile virus. So you have to study each one independently. And that's where gain of function comes in, because let's say, for instance, you were infected with a virus that causes the common cold, okay? The viruses that cause common colds are divided up into groups. And one of those groups is called rhinovirus. You know, rhino, the nose, or rhinoceros, the nose. So those viruses, you get a cold, you get a runny nose, might get a fever, might get some chills, might get a little cough, and you're done. And you might get 1 or 2 a year, or you might get 3 or 4 a year. You might get none, right? Some people don't get colds at all, and other people get them all the time. So that's probably because your immune system is different than everybody else's, because your DNA, although it looks very similar to everyone else's. And oh, by the way, you cannot tell the race of a person by looking at their DNA.
Really?
Really, because there's no such thing as race. And if you want me to go on the air and say that, I will not erase that statement. There is no such thing as race when it comes to biology. There is something called race when it comes to people's perception of biology.
Can you tell someone's skin color by their DNA?
Nope. Not a bit. You can tell if they're male or female.
Is that because we haven't advanced DNA studies enough? I mean,
no, it's because we're very commonly arranged. We look so similar that you can't tell us apart at that level.
I want to say something before we go on about
Please
Coursera. I want to say something in favor of what's the do you know what his Coursera course is called?
I believe it's called Virology 101.
And what's his name again?
Racinello, Racinello. Okay,
I want to say something real quick. So if you are considering doing a Coursera course, especially if it costs money, do it. Because I did one I've signed up for like 50,000 Coursera courses.
You are right. You are right.
But then I found one where I had to pay monthly, right? I had to pay $50. And it was a Berkeley course by a guy called Ludin. I don't remember his last name. It was incredible. It was on synthesizers. And I did it because I had to pay $50 a month. And so when I paid $50 a month, I had to finish it in time because I didn't want to be charged again. And it was the best money I've ever spent. I'm
turning off my camera a minute.
Yeah, sure. I literally signed up for Berkeley for free. It's incredible. And this guy, he's a virologist. He knows everything about virology. I mean, you're getting an Ivy League education for $50 a month instead of.
You got it right.
Yeah. So
that's the deal.
Just do it. Just do it.
Coursera is a brilliant way of spreading knowledge very cheaply. And I can tell you that Vincent doesn't make a lot of money doing it. He does it out of a love for teaching. And his course is very popular. So you can imagine during the pandemic, how many people would like to know what a virus is, right? And he discusses in detail what gain of function means. Okay, so you can, in the laboratory, in cells that grow in culture, you can infect them with viruses. Now, not all viruses infect all cells because some are very specific. And so they can't get in the door unless they have the right key. And that's really the way it looks at the molecular level. There's a slot and there's a plug, and the plug goes into the slot, and that opens the door for the virus to go inside.
Inside the cell?
In the coronavirus family of viruses, on the surface of the virus itself, and you're sitting at your computer, correct? Mm-hmm. If you typed out what does the coronavirus look like under the electron microscope, you will see a beautiful three-dimensional picture of this virus that's gorgeous. And it's got lots of stuff stuck all over the surface, right? But the longest ones are called spikes. Gee, I wonder why they call them spikes. Well, because they look like spikes, you idiot. And so the spike is in two parts. It's like a clam. So when the virus is first produced and thrown out into the milieu of the host, the clam is closed. In order for this spike to function, there's a little substance down over here that's attached to it. It's a sugar. And if you remove that sugar, the clam opens up. It's like taking the beard out of a mussel. When you do that, the clam, the mussel doesn't like that, so it sort of opens up and says, hey, who just did that? You know? When you use an enzyme to take out this little piece over here, this little piece of sugar, the spike now opens and it can now attach to another cell. When that attachment occurs, it's like the key going into the lock. Door opens, come on in. And the rest is history. The virus takes over the cell's machinery to make more virus. It subverts normal biology for its own purpose. It's like throwing a...
So the virus gets into the spike protein by removing the sugar, and then it closes up, and then what is...
No, the other way, the other way.
Oh, sorry, it opens up.
It's closed up. It has to open in order to attach to a cell.
And does that open up a channel so the virus can get into the cell? Is it like a doorway?
It is. No, it's like... it's the difference between having your keys in a wallet and removing the key and leaving it open, exposing it. Okay. The key itself are these two pieces, and they're locked together by this little sugar. If you can remove the sugar, it opens it up, and now it fits the lock.
So
Before that, it does not fit the lock. The lock is on the surface of the next cell. It's not a virus protein. It's a cell protein. It's your protein. So you have to have the right lock and key in order for this virus to work. And that means that you can show specificity of viruses for various cell types and for various animals or various plants. Even plants get virus infections. So
that's why you can't get a plant's virus infection, because you don't have the right spike proteins for a plant. Is that right?
Well, not all viruses have spikes. They have other ways of getting in too. They have other proteins on their side. They are tricky little sons of guns. Because when you look at their genome, it's very short, basically. It only contains about, well, for some viruses, it only contains the message, or the code rather, for 3 proteins.
Okay.
You know, a virus, the simplest virus only needs 3 proteins in order to survive. And some viruses have hundreds of proteins. And what do they do with all of them? And we have no clue, because we can't follow that many proteins. But we know a lot about the coronaviruses now, because everybody just sort of focused on them to try to interrupt this pandemic, and it worked. Okay.
So
and
we've opened up the clam.
And the clam now is a key that recognizes the lock on the next cell.
So the next cell is like drifting around, and then it...
No, it's in a monolayer on the bottom of a plate. It's in a petri dish.
Okay.
So you can grow these cells. That's called in vitro. And they will grow and grow and grow and grow and grow until they reach the edge of the plate, and then they stop growing.
So the cell next to it is like it.
Yeah, exactly. So you take that's a great, I love this discussion because I can actually answer your questions. So if you're a virologist and you want to study how viruses behave, you need 2 things. You need cells and you need virus. Okay, and then you can put them together and watch what happens. So the virus is collected from other cell cultures that you can grow up and harvest. They grow in a fluid. The fluid is their food. You're feeding the cells the food, not the virus. So the cells eat the food, they grow, they multiply, and they stop dividing once they get to the edge of the plate. The entire bottom of the plate is a solid layer of single cells. Now, you want to know, how many viruses do I have in this test tube? They're all the same, and you've just harvested them from another cell, but you don't know how many there are. You can count them, but they're so small that you'll need an electron microscope to do it. So they don't do it that way. What they do is they dilute the solution that has the virus in it. They dilute it 1-to-1, they mix it up. They take that solution, they dilute that 1-to-1, then they mix it up. Then they keep going like this, and they make dilutions of this virus maybe for 10 or 20 different tubes worth. Then they start at the bottom, and they put the whole fluid level onto the plate of cells, and they wait. And what are they waiting for? They're waiting for the virus to settle down onto the surface of the cells. And then they're waiting for the virus to open their clamshells. And then they're waiting for them to go inside the cell. And then they're waiting for this virus to kill that cell and to move on to the next cell. And keep killing cells. Eventually, they'll kill enough cells so that when you look under a regular microscope, you can see a little circle where there used to be cells and the cells are not there anymore. That little circle is called a plaque. And that says that plaque was started by a single virus particle. And the whole thing to do is to do a plaque assay. The plaque assay tells you how many of those little circles do you have in that test tube that you diluted all the way down to almost nothing. You'd swear there was nothing in that tube, but if you start out with 1 billion particles of virus, it's going to take a long time to get to zero.
So we know how long it takes for a virus roughly to infect one cell and get to another. And so
we do. It's about 12 hours.
Okay. So if we wait, let's say 24 hours, we can see the amount of plaque and then have it. And
3 days. We wait 3 days for this.
Okay, 3 days. Yeah.
And that's enough to see a circle. And then what you do is you pour out the fluid and you kill the cells and then you stain them with a blue stain. And the blue stain stains all the cells that are not infected yet. And wherever you have a clear zone, that's a plaque. So you just hold it up to the light and you start counting and you use a magic marker. And you put the marker on the bottom of this plastic dish to make sure you don't count the same plaque twice. It's simple stuff. It's so simple that anybody can do this. I mean, I could teach you how to do this in 2 hours. And by the way, I've never done a plaque assay. So, but we had a whole show on how to do a plaque assay. And I paid attention, of course, and I've worked in a lab all my life, so I know how to do this. So the point is that if every particle that you had in the tube succeeded, then, and if you counted how many plaques you got, that's how many virus particles were in this one tube. But that was diluted 1 to 1, 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 1 to 1. So how many were in the original tube that you started diluting down from? And you can calculate that. It's in the millions. The millions of viral particles per whatever that liquid was.
Silly question. Why do we want to know how many?
I'll tell you why. We're getting to the gain of function part.
Okay, we're getting there. The road to gain of function.
The road to gain of function is paved with, we won't go there. The road to hell is paved with good intentions. That's the expression, right? So the road to gain of function, according to that woman, is paved with not good intentions, bad intentions, evil. We want to take over the world because we want to create microbes that will kill everybody. But wait a minute, we don't want to do that. They'll kill us too. We don't want to do this. No, we don't want to do this. Let's stop. This is not true. This is not true. Even people that are involved in germ warfare admit that there's more risk of developing germ warfare than there is using germ warfare. The people that develop it often get sick and die from their own products. Just like people that make homemade explosives sometimes blow themselves up in the process of making homemade bombs. It's a ridiculous activity no matter which way you look at it. But if you want to understand how viruses succeed, okay? Because viruses don't sit still any more than we sit still. Every time their DNA replicates, there is the possibility for a mistake. And if you count up how many times it has to succeed by adding each of those 4 ATGCs to their own DNA to make a new copy, it's called a mutation rate. And the mutation rate for almost everything is about the same, unless it's interfered with by things like radiation or mutagenic substances. But in the ordinary world, every time DNA replicates, if you take 10 to the 6th molecules, now how many is that? 10 to the 3 is 1,000. 10 to the 4 is 10,000. 10 to the 5 is 100,000. 10 to the 6 is 1,000,000. There's a mutation in the DNA of everything in 1 in 1,000,000 copies. Now, we don't have a 1,000,000 copies of our genome. We have trillions of copies. Our body is loaded with copies of our DNA. So you know that whenever it makes a mistake, it can lead to something bad, like cancers, right? That's your cell in steroids, as they would say. Your cell now doesn't know how to stop replicating, and it creates a problem. And it's very common, it turns out. And some tumors grow and then burn out, and some tumors continue to grow and then they kill you. Okay, that's your problem. The virus problem is, I don't replicate very well in that cell. Well, guess what? Just wait. And maybe over time, a random mutation will lead to a better fit of the virus for whatever it's learning how to do. And it learns how to do it by just making more of itself. So what if 1 in 1,000,000 virus particles learns that lesson? Then when that one virus particle, lucky enough, hits a cell and replicates, from that point on, it could outcompete all of the other viral particles that it had before it. So it becomes the dominant virus. And eventually it replaces all the virus with itself. And if you look around the world at this coronavirus and how it's behaved, that's exactly what's happened. Let's say it started out in Wuhan, China, at a live animal market that people buy live animals and take them home and slaughter them and eat them, and that's the way it is. That's their culture. That's not ours. I wouldn't say that. We tend to do things like that, crazy things. People will eat anything that swims, flies, crawls. It's true. Okay, so this virus started out with an archetypal, we'll just call it the original sequence of its DNA. It's an RNA virus, by the way. So it's already made the message for itself, and that's what it's going to put into the next cell. Okay, so it's an RNA virus, not a DNA virus. It's an RNA virus, which is why this vaccine works so well, because the RNA virus, we can copy it and we can disarm it. And so we're not giving you the whole virus when we give you this vaccine. We're giving you the entire code for just that spike. It turns out if you can stop the spike from functioning, you can prevent I'm not going to call it alive because it's not a life form, but you can prevent an infectious particle of virus from infecting a cell by making an antibody that interferes.
I'm super confused. Okay, so
I know you are. Of course, I'm confusing you on purpose. Okay. I want you to ask questions.
Okay. So
what's the point that you didn't understand?
So the spike that we're talking about is in my cells, right, and not in the virus.
No, it's on the virus. It's a virus.
On the virus. Okay, got it. Okay.
It is a virus thing. Remember when I asked you to look up the structure?
Oh, okay. I thought
that doesn't have any host things. That's all virus.
Human cells don't have spike proteins?
They do not have spike. They have the lock.
Oh, they have the lock. Okay, cool. So
and the spike is the key. That's simple to keep track of.
So how does
you want to open a door? You need a key.
Okay, but what process is removing the sugar then? Because if the virus already has key.
Most clever question. Who removes the sugar? Viruses can't do it. So I guess you did it.
Is it like a bait? Is it like my cells are like, oh, sugar, delicious?
Well, no, but on the surface of your cell, believe it or not, there are enzymes. Those are other proteins. And an enzyme is a machine that catalyzes a reaction. And this reaction removes furan. It's maddening when you think about how many advantages this virus has taken in order to gain entrance into a cell. Yep. It has to get the cooperation from that cell in so many different ways. So that's all done by random mutation. All right? Okay. This is an ongoing process. Yeah. Every day, every way, we're trying to make new versions.
And so the vaccine is coming into my cell, and it's giving me the protein it's giving my RNA, which is leaving my nucleus and coming in and going into the cytoplasm. It's giving it the protein for the spike protein of the virus.
I will
Okay.
correct just a little bit of that.
Yeah.
Okay. So the vaccine, what is that vaccine anyway? Okay, it's a piece of RNA. It's not a protein. It's not the spike. It is the message for the spike. So we can copy that message from the virus because it's got that message in its own genome. And we can take that little piece, which is non-infectious, and we can make a bunch of it. I mean, a ton of it. And then we can encapsulate it. And why do we have to do that? We have to make it look like a virus. Otherwise, it won't be taken up by a cell. The protein RNA complex will not occur because if you just injected RNA into the blood, we have an enzyme called RNase. What? That actually chops up pieces of foreign RNA that come in. So that would ruin the whole idea about a vaccine because the RNA wouldn't even get a chance to get into a cell before we could start making spike protein. And that's the whole idea about this vaccine. The vaccine wants a cell, your cell, normal cells, to make just a little part of the virus, not an infectious part. Well, because
you want to make the spike protein.
Well, I'll tell you why, because it's the key that the virus uses to get into cells. So if you can make an antibody against that key that floats around in your blood, when new viruses pop out of a cell, the antibody instantly gloms on to this spike.
So is the antibody, okay, so the virus.
The antibody is a weapon against spike.
So the virus is coming into my cell, and it's
going to try. It's going to try.
Oh, sorry. The vaccine comes into my cell, and it puts some RNA into my cell. Is that correct?
Correct.
Okay.
Just a little bit.
And then what does that RNA do in my cell? So that RNA
makes spike protein.
Makes spike proteins on my cell. So my cell grows spike proteins that it didn't have before? Okay.
That's correct.
And then
Then what happens?
Yeah. Then what happens?
Then what happens?
Do they hit other cells that I have? And then
No, I'll tell you what happens. You're a complicated organism.
Yeah.
This is more than a 2-hour podcast, by the way.
That's fine. That's fine. As long as you're okay with that. Yeah.
I've got to eat. But we can make this part 1. I could come back.
Okay, great. Yeah.
I would love to. So
yeah.
So if I can get you to understand this part, you'll see why we need to do gain of function.
Yeah.
All right.
Yeah.
That whole conversation's about that. Right. So we've figured out that if we can make an antibody, and what is an antibody? An antibody is made by an immune cell. You have an immune system, and everybody talks about it. Eat this, don't eat that, don't eat this, don't go near those. It'll spoil your immune system. So your immune system is on red alert. Okay, and that means that it's geared up in many different ways to survey your body to make sure that you do not become infected with a bacteria, that you do not become infected with a protozoan, that you do not become infected with a worm, that you do not become infected with a virus. Okay, and now each one of those things that I just mentioned takes a different portion of your immune system to address the issue. So viruses have gained entrance into our bodies since there were bodies, and we have remnants of their DNA in our own DNA. We have long-term repeats, long-term sequences that said, you know what, this sequence looks like a virus. It's not a full virus, so it can't replicate, but it was at one point, and it got into our DNA, and it just left part of itself there. So a great deal of our own DNA consists of ancient exposures to virus infections that occurred over our history of evolution. That's remarkable, but that had nothing to do with your question. Your question was, what good is getting RNA that encodes the spike protein into a cell? Because what we want to do is we want to create a foreign protein that the immune system can recognize that says, oh, wait a minute, there's a virus loose. I know it because I can detect a spike. Well, what the immune system doesn't know is that That's all it's detecting. There are other things. If you look on the surface of the virus, you can see lots of other things that are there. But the one thing that our immune system seems to recognize the best is the thing that sticks out the most from the virus. The most obvious thing to go for would be the spike. Okay? So when we want to make antibody against just spike, we would never use a whole virus because all those other things would interfere with the process.
Mm-hmm.
So we have cleverly figured out, using our own brains, took long enough, but we did it, is to make a vaccine which consists of the RNA coding portion of SARS-CoV-2 that makes the spike protein only. You don't get sick from that because the virus doesn't replicate if you only make that protein. You have to replicate a virus and then it kills the cell. If you replicate a portion of the virus, all we do is just handle it as extra protein and we eventually degrade it and it's gone. What happens to the spike is interesting because we've also engineered a little piece in there that allows it to be expressed on the surface of the cells that got the RNA vaccine virus construct to begin with. And it could be a muscle cell, it could be a fibroblast, it could be where you inject it into your muscle. Wherever that is, that's where it gets into cells and then they start making spike protein. Oh, it's like giving the protein precursor. You don't give the protein because that's too much effort. Let's let the body make it. And we can make it much more efficiently and we can make it correctly. And on those cells, the immune system now comes along and says, hey, oh, there's a virus over here. Come on, get busy. And you do two things. One is that you make antibodies. And the antibodies are the liquid part of your immune system that guards against invasion. But lucky for you and lucky for me, particularly because I'm much older than you are, I don't make antibodies too well anymore. When you get older, you don't make antibodies as well as you should. That's why we got boosters for the older people, and not for the younger people. Not right away, but everybody now is getting a booster shot. But the point is that you've got another set of cells. You've got one set that makes antibodies, and you've got another set that kills infected cells directly. They're called T cells, killer T cells. You've got a third set that's called natural killers, but that's a different program. Do you ever watch cooking shows on television?
No, I hate cooking shows.
Oh, too bad. Oh, you don't like to cook?
I love cooking. I love cooking. I'm a good cook.
Oh, you do? Well,
I don't want to watch people cook.
Well, there's a cook, there's a chef on television. His name is Alton Brown. And I love him because he's a chemist.
Oh,
and he gives you the chemistry of cooking.
That's cool.
And he'll get to a subject like we just got to, and he'll say, but that's another show. I'm not going to say about natural killer cells. So you've got antibody producing cells that interfere with the key coming in contact with the lock. It actually is a big molecule, so it sticks on the outside. It can't get in. And that means the virus can't infect the cell, and therefore eventually it becomes degraded and it's gone. That's a good thing. And you can measure how much antibody each vaccine produces, and some people produce more than others for the same dose. That's interesting. Why is that? Because we're all a little bit different. And therefore, we behave a little bit differently. Some people get allergies, some people don't. Some people can't eat shellfish, and other people love them. Like I'm a shellfish lover, I love them. But I have a lot of good friends that'll die if they eat shellfish, because their immune system doesn't recognize it as something normal, and it starts to attack its own body. So that means that the shellfish contains a protein that looks a little bit like your protein. Now that's called autoimmunity. When you start making antibodies against your own tissues, that becomes a problem with that. But as Alton Brown would say, but yet that's another show. What we really want to know is what happens to this RNA molecule that encodes the spike protein only. It's the message for the spike protein. What happens in the body? And two things happen, both based on the fact that all the cells that received that RNA molecule start to make spike protein. They've got the code for it. We might as well make it. What the heck? And they do. And we can express it onto our surface so that the surface of a cell now has lots of little spikes, just like the virus. It looks just like the virus. Look, the virus is over here.
Mm-hmm.
And so the immune cells come in and they copy a portion of the spike, believe it or not. They then go and give that little message to another set of cells. They say, yummy. I can make an antibody exactly the fit for this protein. And when that happens, you start to prevent virus from entering cells. Your antibodies actually stop them because they stuff up the hole for the key.
Wow.
You can't get a virus in that hole because it's already got something in it. How did that happen? It reacted with the surface of the spike and it tried to stuff it in and it doesn't work. It's the wrong fit. All right, so say, for instance, you're an older person and you're not making antibodies as well as you should. You still have another line of defense, and those are the T cells that kill infected cells. So if, for some reason, there's a breach of immunity because your antibodies didn't work out too well, you can still have this fallback position. Your T cells can still kill cells that are infected at a bigger rate because you've got the vaccine. You've alerted the immune system in general. You can hear the sirens. Every immune cell can hear the sirens ringing, and they all respond differently to those sirens. Red alert, invader, invader. You can hear it now. Will, we're in trouble, lost in space. The whole idea then is, how did this virus ever get involved in us anyway? Now that's a gain of function. So if you have a SARS-CoV-1, which there is, we just called it SARS. It's called severe respiratory syndrome. And it was in China, another Chinese epidemic. And it was traced back to civets, wild animals that are caught and then raised in captivity for food. But somehow it disappeared. It came up, it killed a few people, not just a few, thousands, and then it just went away. And we don't know why. Maybe because people stopped eating civets, maybe because they weren't eating horseshoe bats anymore, because they were warned, that's where this virus is coming from, don't eat those anymore. Maybe that worked, we don't know. There's a second one called MERS that infects camels and camel herders. And some people have died from that too. That's SARS, MERS. It's a coronavirus. It's related to the virus that we've got right now in this pandemic. And it too has sort of smoldered. It hasn't really grown into a pandemic. Camels are not found everywhere. And so maybe that's the reason why MERS is not going anywhere. But this virus, SARS-CoV-19 or SARS-CoV-2 19, that virus resembles a lot of common cold viruses among animals. They've got, if you compare the sequence of those ATGCs, and you line all the coronaviruses up, you can find very common regions in all of them, including the spike protein. Like almost all the coronaviruses have spike protein. Not exactly the same, but close. So I can now answer your question really simple. This is an easy, simple answer, and it's the right answer too. Wouldn't you like to know where the next SARS-CoV is coming from?
Where?
Oh, that's right. Where? How are you going to find that out? Oh, let's just think of this as a problem of you get a whole bunch of sentences of a book, and they all look the same, but they're not the same. And your job is to take those sentences and stretch them out. And you've got 20 of these or 40 of these sentences. And when you line them all up, you start with the same words for all 40, and you continue those sentences across to the end, and you start to read them to see if they're exactly the same. You can line them up so you get one that's 99% the same. The next one is 96% the same. The next one is 20%. No, not 20. The next one is 80% the same. The next one is, so you get a row of sentences that are a percentage identical to the one that you're comparing them to. Okay, sentences, just words, right? So that's all a genome is just a set of words that the body uses as a chemical to transfer information. That's what a sentence is. It's all about information, right? So it's not different. So we can take SARS-CoV-2 and we can sequence it, A, G, T, C, A, G, 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 G. And it has, you can look it up, it has like, I think, I won't guess because then I'll reveal my own stupidity. I forgot the number, but it's got a lot. Okay, but we have machines that do this. They're called DNA sequencers. It's very easy to do it. You convert the RNA to DNA. Remember, the RNA is the message. So now in a machine, we can make a replica of the RNA that turns out to be actual DNA. Now we can take that DNA and we can make a lot of it. And then we can take the sequencer, the DNA sequencer, and we can tell you exactly the way the A, T, Gs, and Cs are ordered. And there are millions of them, millions, okay? Every SARS virus has a different set of spike protein RNA messages. So we would like to know which one is 99% identical to the one we've got, because all you need is one more change and now we can catch it. So what happens if the one that's 99% the same now succeeds by random mutation to make a version of itself that looks exactly like the SARS-CoV-2 that we're connected with? Do you know what that's called?
What?
Come on, you can say it. What is this whole show about?
Come on,
you can do it, Anna, you can do it. Yay! So instead of waiting for nature to do it, then we're in deep, you know what? I will use the word deep yogurt, but you can imagine what I would have said because I'm a parasitologist. You want to edit the word shit out? Go ahead.
No, it's
so,
fine.
no, please don't.
I won't.
So nature does a gain of function every time it replicates the virus. It's ongoing.
Okay. Oh, okay.
There's no way to prevent it. So this stupid woman that thinks that you can prevent it by preventing graduate students from working on gain of function, what a dumb, she doesn't know anything. And you can tell her I said so.
Okay.
Because viruses do this so much better in nature than we can do it. Eventually, the gain of function will be succeeded by this parasite. And we will catch the next version of this virus with our pants caught down around our knees. And we will be in the same position we were in 2019 when this thing first started.
Okay.
Unless we go into nature and we collect animals that are common to regions where this outbreak is suspected of coming from. And we isolate their SARS and we compare them to the SARS-CoV-2 that we know. The closest one we can find so far is in a pangolin. You even know what a pangolin is?
Yeah.
Oh, good. It's considered a delicacy and it's prominent in Chinese medicine. Neither one is correct, but that's their preference, not ours. Their SARS-CoV-2.
Have you eaten a pangolin?
No, but I've seen them.
Okay, yeah.
In Africa, I saw a pangolin in Africa.
Wow.
A live pangolin. So this pangolin RNA, which encodes its own spike protein version, is 78% or something like that similar in the code region all the way across to the one we have. So that's a lot of changes that will have to occur before it can actually look like ours. It might be several hundred or several thousand that have to fall into place before the pangolin can serve as a vector for this virus to people. Because people catch pangolins and they eat them and they destroy nature, basically. So better to take our virus and deconstruct it and see how far back you can go before it loses its function. So that's called loss of function studies, and we do lots of those studies all the time. We would like to know, how many changes do you have to make in the RNA of the virus so that it does not infect cells? Right?
Yeah.
Okay, so that's called loss of function. So now we could take an animal SARS-CoV and start making changes to say, how many changes do we have to make in this virus in order to get it to replicate in human cells in a petri dish? That's the way the experiments are done. They don't use animals, they use cells, and they use facilities that prevent the spread of this virus. They're called P3 and P4 facilities. They're very safe places to do these researches. Not that you can't make a mistake, but it's very, very rare that that happens.
Okay, so for gain of function, we might take a pangolin cell, and we
Right.
might so it's like 78% accurate, and we might have a billion of them, like in a test tube,
Correct.
like you said before.
Yep.
And we might have it spread around to a bunch of different cells and see kind of how fast it spreads, and also we might see how much it changes in that time. Like if it's 1 in a
could.
million and there's a billion of them, then it's going to
You
change.
could, but
1,000 times.
that's a long-term experiment, right?
Okay.
That sounds like a lot of work.
Okay.
We don't have to do it that way.
How do
We
we?
have easier ways of doing this now. We could actually artificially make a virus.
Why would we do that though? I mean, if
So
we're
then
looking-
we can
oh,
insert all the changes we want artificially. We don't need a cell. We can do this by a chemical reaction.
yeah.
So Dr. Vincent Racaniello, who you're now familiar with by name, became famous for constructing an entire poliovirus particle from scratch.
Hmm.
It's like making a scratch cake.
So this is like
For you.
CRISPR technology?
No, way before that. It's a chemical process. You take A plus B and you get C.
Yeah.
Take C plus D and E and you get F plus. He constructed, based on the code of the entire poliovirus genome,
Yeah.
he constructed in a test tube the genome that then infected a cell and made poliovirus.
Mm-hmm.
Genius.
So why are you making? So are you
Proof of concept. In his day, he's an older person. So in the days of old, you had to take baby steps before you could run. So we just wanted to know if the DNA alone of this virus is enough to make another virus particle, or do you need something else? And the answer was, you don't need anything else. You just need
Right.
that.
Okay.
And he was the first one to show that. Okay, so now if you can do that, you can manipulate the order of things. In other words, you can take a sentence in English and change it to Italian, or you can change it to French, or you can change it to Icelandic. If you do that, no one will read it, not even the people in Iceland.
Nope.
So, because it's a very difficult language. The point is that you can manipulate in vitro, it's called in vitro, the sequence of DNA without using cells as the way of doing it, which makes it easy to do. So then that's what's going on now. And they're studying how much change do I have to elicit in this genome to get it to infect a human cell. And that's called gain of function. And that's considered dangerous because once you get it into a human cell, how could you prevent it from spreading all over the world and killing everybody? Well, we've done this lots of times.
Okay. So I want to stop you there. I want to recap. I'm trying to.
And then we're going to be done because I have to go eat.
Okay. Do you want to finish in how much time do you have?
How much time have we used so far?
We've used 1 hour and 27 minutes.
Oh no, we have lots to go then. Come on,
Okay,
go.
do you wanna-
You said
do you
2
wanna
hours,
finish-
right?
You want to finish in half an hour? Yeah, perfect. All right, cool. So I'll
Okay.
let you know when we're like 5 minutes away. from-
Okay, no, I don't want to stop you from asking questions.
No worries, no
Because I
worries.
think your listeners will have the same questions.
Yeah. So we're gonna take a pangolin cell and we're gonna change it. We're gonna see how
Not
much
a
we
pangolin
need-
cell. We're not going to take the pangolin cell. We're taking
Oh,
the pangolin
SARS virus.
COVID. Yeah, that's right. The SARS virus.
Yeah.
It's not
Okay.
the cell of the pangolin. It's the virus of the pangolin.
Okay, so we're gonna take a pangolin SARS virus and we're gonna
Yep.
change it as much as we can. So we're gonna change it until it can infect a human virus and until it
Correct.
can infect a human cell. And
We don't know how many changes that would equal.
And we don't know which changes too, because like 76.
You could line them up. You can line up our SARS-CoV-2.
Yeah.
And you can line up the pangolin. Now,
but only like
you don't have to change everything.
Yeah, you don't, and then we can figure out which changes will infect us. Is that why? Is that one of the reasons? Okay.
Correct.
So we're trying to figure out
how will the virus have enough time within our lifetime to evolve through mutation to a human pangolin virus rather than just a pangolin virus. Because what we haven't identified yet, and we would love to identify, is the animal that has a 99% identical SARS-CoV-2 virus. We haven't found that animal. We looked and we looked and we looked and we
Why
looked.
do we want to find that animal?
Because it only needs one more change in its our virus.
Oh, and then we can get rid of it before it happens.
Unless, of course, it requires more than, well, not unless. It's probably infectious way before it's 99% identical. And I'll tell you why I think this is true. Because for other viruses, like, for instance, West Nile virus, the virus that originally infected people came from Israel. We know this for sure because Israel had an outbreak of West Nile virus first. In that same year, West Nile virus was introduced into the United States by tourism to New York City, to the zoo in New York City, by a little kid. We don't know his name or her name, but they had this virus infection and they spread it to mosquitoes, and the mosquitoes then spread it to crows and exotic birds inside the zoo. And so all we had to do was isolate that virus and compare it to the West Nile virus outbreak that occurred in Israel. And it was 99% identical. And there are lots of West Nile virus versions out there. There are 23 different West Nile virus versions, and none of them come even close to this one. So we know exactly where this virus came from, and we know what happened after that. Now, if you look at the West Nile virus outbreaks in the United States today, they're totally different than they were even when they first were introduced. They're like 88% or 89% or 76% identical, but they've all changed because mutation. Remember, mutations are 1 in every 10 to 6 replications, right? So that's a lot for a virus, not for us. But a virus makes 10 to the 20 or 10 to the 19th particles in a person that's sick. That's a lot more than 10 to the 6. You imagine how many mutant viruses there are. It's a lot. I mean, the number is huge.
Yeah.
So that's the numbers game.
Okay.
So gain of function means how many changes do I have to make in the genome of an organism before it becomes infectious for us? Period. It's very simple. And people think that's dangerous research because once we release the genie from the bottle, so to speak, you've got a new virus to worry about that you never had before. Not realizing, of course, we've been doing this for years, and nature does it all the time. So why should we be different than nature?
Is it possible that we could, with gain of function research, create a virus that is more dangerous than previous viruses were?
We'll never know.
Because to me, it seems like
We'll never know. We'll never know the answer to that question because we'll never release it, and you'll never find out.
We'll never release it on purpose. So now I'm getting
on purpose. Now you're getting conspiratorial on me.
Well, it's no. I'm not conspiratorial. I just know how much human error is a factor.
No, of course you're allowing for that.
You can break a petri dish full of a dangerous virus.
Give you an example. The example that I will give you is smallpox.
Yeah.
So we've known about smallpox a long time. We've been able to raise smallpox in the lab. We've been able to store it. We've been able to ship it. It's very infectious. People die from it. And yet, there have only been 3 laboratory accidents with smallpox virus ever.
We know of.
2 of them occurred. No, we know this for sure because smallpox is gone now. There's no more smallpox in the world. Nobody catches smallpox because there isn't anymore. It's one of the few viruses that we actually got rid of. But people are still secretly working on smallpox virus. And you would know it in a minute. You would have people infected. They would spread the virus. People would become more infected. And eventually say, there was an outbreak. And you could say exactly where that occurred, okay? So that's what happens when you get an accident in the laboratory. So there was a laboratory accident recently in England about 10 years ago. That's recent, where an investigator made a mistake. The virus actually escaped from his laboratory in a stream of liquid. He threw it down the sink, which is stupid, and the sink was leaking, and the leak transferred down to the laboratory of another person on the floor below, and she caught smallpox and died.
Wow.
That's death number one. Death number two, he committed suicide as the result of his act. Those are two deaths from smallpox that were released from a laboratory accident. That's the kind of thing you're dealing with here. So the question that you've asked is valid, but we're now looking at the 1918 flu virus, which killed 2% of the world. And we're asking the question, why? The flu viruses that we have today don't do that. We have never had an epidemic as serious flu influenza as we did in 1918. It was called the Spanish flu, but it was a horrible name to give it because it was everywhere. So now we've dug up, how do you even get the virus to look at it to see what it was like? And it turned out to be very easy. In Norway, they buried a bunch of people who died from it in the permafrost, and their bodies were preserved. And the virus was still infectious that they could isolate from the bodies when they dug them up. And they then infected cells in vitro in the laboratory with that virus, and they sequenced the entire virus. And now they know exactly the region that changed to make it the most dangerous flu virus in the world. Now, isn't that worth knowing?
Yeah, but
they didn't do any gain of function studies. They just wanted to know why this virus was so dangerous.
Okay.
Now you're asking, can you make it even more dangerous?
I think they could.
You kill off 100% of anything?
I think they could. I think they. I mean, I don't know.
You do, because you don't know anything about DNA. That's why.
You're right. You're right.
Or evolution. Evolution doesn't work that way. That's not how evolution works.
It's always a percentage of people who are different.
Yeah, there always got to be a mutant out there that could resist everything. I'll give you the best example that you could probably know that you could relate to. The AIDS virus is a horrible virus.
Yeah.
It's slow. It's very difficult to detect before it's already caused its damage.
But there are people who are immune to the AIDS virus.
That's right. Totally. And they know exactly why, too.
But, but AIDS still kills 99% of people.
Oh, no question. No question.
So
what if you could make a vaccine based on the difference between the ones that lived and the ones that would die from this? Would you make a vaccine?
Right?
Well, we were trying. We're
trying, yeah. But here's
we haven't succeeded yet.
I mean, I don't know my stance on gain of function research. I don't think I know enough about it, but isn't it possible?
Neither does anybody else. We haven't done enough. We haven't done enough of it.
Yeah. But I mean, devil's advocate, isn't it possible that somebody could potentially create a new AIDS virus? And, this is the Swiss cheese model. Somebody's got to fuck up multiple times here. But what if they make
I'm turning off my camera for a moment.
Oh, yeah, sure. No worries.
Pausing, as it were.
Oh, you want to pause the video?
No. I'm going to just keep talking. That's
okay, cool.
Go ahead.
So what if somebody creates something like an AIDS virus that has a 99% kill rate, and then also
whoa. How would they do that?
Through
Tell me how you do that.
They take a pangolin DNA. A virus, a pangolin.
But you have to know,
and then they
say, to know how the work works. You need an animal.
Okay, let's say they take the AIDS virus from a monkey, the rhesus monkey, and then they say
that's a simian virus.
And then they say, how close can we make this AIDS virus from this rhesus?
They've been trying that experiment for 25 years without success. It's a difficult virus. It's got lots of parts. It's got huge numbers of genes. It's got a complicated life cycle. It doesn't revive around a spike. It's got two different parts to the virus that actually need to attach to the cell in order to get a cell to take it up, and it only infects B cells.
Do you think there's almost no chance that we could accidentally release, through gain of function research, that we could accidentally release a dangerous virus to the world?
You mean like Dr. Mengele is sitting in his laboratory in his basement and
because there's two different categories.
Look at all the technology that's necessary just to work on viruses.
Well, there's two different categories here, right? Dr. Mengele would be an intentionally malicious actor. So obviously, correct. I think we should all be against intentionally malicious actors.
Yeah, but you're not going to stop them just because you're against them.
But yeah,
Putin would be bombing the hell out of Ukraine, right?
But gain of function research, I think, would be categorized as something different, right? Gain of function research is not intentionally making viruses to kill a certain population. This is, I would say, it's something like manipulating viruses in order to solve the problem. And the question is not, should we be intentionally doing genetic warfare? The question is, should we be researching viruses and changing them in order to figure them out better? Is that correct?
Yes, but you're asking the same question as someone would say, well, why don't we only write sonnet poetry? Why do we have to write iambic pentameter poetry, which is more difficult to pronounce and not a lot of people read it and that sort of thing?
Yeah.
Why would anybody try to prevent anybody from exploring natural process? Because that's what you're doing.
Well, the answer is because
science is curiosity driven.
Iambic pentameter poetry doesn't spread smallpox to people, and I'm not against it, but I'm arguing devil's advocate here.
Neither does gain of function research, because in the most cases, they're not even studying human viruses. They're taking a fish virus and they're studying that as to whether or not it can be more infectious for a zebrafish or not. But that's not going to hurt anybody. So if you limit it to non-human viruses, let's say that you were the oversight board for the National Institutes of Health, and you're a bunch of really studied scientists, you've all done bench research, you know what it's like, and you know it's necessary. And you could say, well, I think we'll restrict gain of function research to non-human issues.
Is that what they do?
No, but if you say that, then what happens? You learn all about nature except the part that you're really most interested in.
Yeah.
What will the next pandemic look like? Where will it come from? And how can we prevent it?
Yeah.
And if you don't do gain of function research, then you're never going to find out that answer.
So it's your argument that gain of function research is
you will live by the seat of your pants from nature supplying that answer for you, and the answer comes too late.
So I think a lot of the things that people are against in gain of function research is, I think they're conflating these scientists who are trying to prevent the next COVID with.
You are right.
With Mengele.
You're absolutely right, because Mengele could not have done that. Yeah. In fact, what you're suggesting has never been done. We've never created a super pathogen for anything.
Have you heard? I mean, I'm sure you have. Everyone has heard. Do you? This is provocative questions. The next 20 minutes, I want to ask you a couple of provocative questions. So,
this hasn't been provocative?
No, not enough, clearly.
Fair enough.
What do you think the odds are that COVID was made in a lab?
Where do I think that's ridiculous?
Why is it ridiculous?
Because no one has ever done it for anything else. Why would the Chinese suddenly succeed where everybody else has failed? They're just as competent or incompetent as everybody else.
Because if I were Xi Jinping,
that's why. That's my answer.
If I were Xi Jinping, I would say.
By the way, if it escaped, let's go back to the laboratory. Okay. Come on. You remember the story I told you about smallpox?
Yeah.
Why didn't that happen to the laboratory workers?
Because it dumped down the drain.
Nobody died in Wuhan laboratory.
They got
SARS-CoV-2.
The Wuhan laboratory people got COVID though, didn't they?
Of course. They went out in the street and they bought food and they went home and ate.
They died, but it wasn't because COVID isn't very deadly. I mean, they were youngish laboratory workers who were
Not the
fairly
senior
healthy.
workers.
I mean,
The senior workers could have easily got this and died from it.
yeah, but I mean, it really doesn't have a huge death rate. I mean, it's
No,
statistically.
but you're going to have to answer that question first, because when they went back in the laboratory and they swabbed everything and they looked for the virus, they
Yeah.
didn't find it. But
Well,
that's because
you can-
maybe the
you can
Chinese
clean.
cleaned it
I
up
mean,
just before we went back to look.
You just
Do you
need some
think that might have
isopropyl.
happened?
It's not very expensive.
That really feeds into the conspiracy theory about all this.
I just don't.
Because
I just
people
don't
want to
like-
blame it on something. No, you got to blame it on something. So
Okay,
what do you blame it?
here's my idea. I don't know. I have no idea, right? So I kind of in the
Serene,
middle of-
Serene, you're far from serene. I must tell you, you're
That's
a very
okay.
active. You're a very active person.
Okay, that's good. I like being active. I'd rather be
At
active
the end of
than
this
passive.
podcast, I'm going to share my screensaver with you to show
Okay.
you. It's called Serenity. You'll love it.
Okay.
You'll absolutely love it. You'll want it as your own screensaver. So, but keep going with the Wuhan laboratory hypothesis.
Okay, so I just want to make clear that I don't have an opinion here. So like I mean I have many opinions, but I don't know which one is right. So I
What?
just want to argue. I just want to ask you something
Sure.
and see how you respond.
Go right ahead.
If I were Xi Jinping, I would probably say if I had a bloodthirsty power, if I was a bloodthirsty, power hungry person, if I wanted to take over the world or have control, I might say, hey, let's take the cold and let's make it deadly. And I don't know. I mean, I think it's possible that the Chinese government is operating more as Josef Mengele than, gain of function researchers who are.
I have a
who want the
question
best
for
for the
you. I
world.
want to
Go
stop
on.
you right there then.
Yep.
Okay. So, in order for that to work, it could work if we would like China to take over the whole world.
I don't want China
By
to take over the
inventing,
world.
no,
Yeah.
but if China wants to take over the whole world,
Yeah.
right? They want to take it over economically. They
Yeah.
don't want to kill the world. They need customers, for Christ's sakes.
I don't think they did it on
Think
purpose.
about that.
So if they did
No.
it,
Think about that. So let's say that they thought, could you create a virus that kills everybody?
Hmm.
Do you think you could create a virus that kills everybody?
me personally, no, and I don't think anyone
I
else.
don't either.
can,
After
based on
hearing
this
what I've told
conversation.
you,
Yeah, no.
after hearing,
No.
I mean, how complicated it is just to study the virus.
Okay.
Right? If we could create another flu virus identical to the 1918 virus, you know how many people we would kill?
Well, it had
I don't know.
Didn't you say it had 100% death rate? Not everyone.
No, we killed 2% of the world.
Oh, you said 2%, not 100%. Oops.
Yeah,
Okay.
you have to listen more carefully. Nothing ever has killed
100%
everything.
death rate. Okay, I'm
Okay?
gonna play
Except
the
one
part.
thing.
What?
No, except one thing. No, I'll give you the virus. You want a virus that does that? There is one virus that everybody knows that if you become infected and you don't get treated, you will die.
Rabies.
Okay, there you go. And you don't know anything about viruses, you've been holding back. Don't hold back. Why
So
would you
I'm-
ever have to manipulate any virus? Just give everybody rabies. Except the Chinese army, all the Chinese merchants, all the Chinese people. Imagine an entire country that was vaccinated against a single virus. Every single person had to be vaccinated. Period. You cannot escape. And now we're free to release it to the rest of the world. And I'll conquer the world. And no one will be left alive except us. Yay! Nobody thinks like that. Nobody. Not even Putin. You know why Putin is attacking Ukraine?
Why?
No, I asked you first.
Um,
I'll give you 3 guesses. Money, your microphone. No,
yeah. Oh, my
money.
microphone. Yeah.
That's okay. Money, no. Territory, it's got plenty of territory. Siberia, do you know how big Siberia is? It's got 1/3 of the
But
land
Siberia
mass
is
of the-
useless. Siberia is totally useless.
What,
I mean, it's
total,
it's
well, I
cold,
wouldn't
you
go
can't
that
grow
far.
anything. You can't grow much. without
What?
growing anything
You
else.
can't what?
Oh, okay. You can grow indoor farming. You can use indoor farming.
No. Go back. What do they grow in
cold.
Ukraine?
They grow. What do they grow in Ukraine?
I asked first. What'd you have for breakfast?
I didn't have breakfast today.
What'd you have for
I
lunch?
have eggs a lot.
Me
I eat
too.
a lot of.
I do that too.
I have.
What'd you have for breakfast? What'd you have for lunch?
Oh, I haven't eaten yet. It's only 10 a.m. I had.
Where are you located?
Sydney, Australia.
Get out of here!
Yeah.
Are you kidding me?
I grew up here. Yeah, I grew up in Australia.
You did not. You don't even have an accent.
I know. I'm American by birth, and I spent a lot of. I grew up in Australia since I was 7, and I've always had the accent.
Guess where I spent 6 months on my sabbatical?
Was it Australia?
Yeah, Melbourne.
Where oh, I love
Melbourne.
Melbourne. I went to university.
I do
there.
too. Did you really? Where?
Beautiful. Deakin University. It wasn't. I didn't get good grades in school. I was not a high performing student.
What?
Yeah. So it's like
Sydney, Australia. You're a Sydney sider.
Yeah.
I've been to Sydney lots. I spent a lot of time in Sydney. It's a great place.
It's beautiful.
You just got a big flood.
Yes, we did. We weren't affected. Well, we have a farm that's a little an hour away, and that was affected. The bridge was cut off. Everything. We have alpacas, and the alpacas are very upset. They don't like being rained on constantly.
Yes, you have an interesting spider that inhabits the same area that you do. Also, funnel
yes,
web.
the Sydney funnel web. Yes.
Ow! Oh, they're gorgeous.
Yeah.
So let me ask you the question, then. I come back. What do they grow in Ukraine that Russia can't grow? What'd you have for lunch? You didn't have lunch. What are you going to have for dinner?
Is it some sort of?
Starts with W.
W.
Yeah, it starts with W.
Water.
Wheat. 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 Wheat.
Yeah. Okay.
Wheat.
Okay. Wheat. Yeah. Okay.
Wheat. Russia can't grow wheat unless they have Ukraine.
Okay, wait. I want to. Okay, go on.
8 years ago, they had Ukraine.
Yeah.
Ukraine voted to get rid of Russia because they were parasitic on their farming. They were taking all their food. You start with wheat. Wheat is a storable product, right? So that's what that war is all about. It's about resource management. It's horrible.
I have a question though. So I'm going to
wait a minute.
Okay. Go on.
So if rabies is the only virus that kills everybody,
yep.
Why would you want to kill anybody else with another virus that doesn't kill everybody? What's the purpose of killing people in order to take over the world? You don't do it that way.
I have an answer though. And
Tell me your answer. What's your answer?
This is from frequenting conspiracy theory forums, and it's not something I believe.
Oh,
but
God.
It's
By the way, I'm never going to go on your show again.
You already said you would. You can't take it back.
No, it was a conspiracy. I took it back.
Okay. So I don't believe this conspiracy theory, but I want to ask on behalf of conspiracy theorists.
Don't do that. No, I don't think you're allowed to do that.
No, I'm not.
I want to talk to you. I don't want to talk to your conspirators.
No, but
So you don't believe it. You're going to give me something that you don't believe. Tell me why you don't believe it.
It's not that I shouldn't say I don't believe it.
You can say you don't believe it.
I hang out in the realm between belief and non-belief a lot.
Yeah, I kind of figured that when I agreed to be on your show.
So I don't know what's true, but I have questions. They've raised questions that so you said
can't answer.
That I can't answer. And that's why
I raised more questions that you couldn't answer, but now you can answer them.
Well, that's the
value of teaching.
You got diverted. So let me ask you. So you said, why would Xi Jinping want to kill everyone? Maybe he doesn't want to kill everyone. So what some people say was that China has an aging population. For every one person, because of the one-child policy, they have 2 parents and 4 grandparents that they need to support financially, which is terrible. You can't be an entrepreneur if you have 6 people potentially to support. You all your money is going to older people. So
how do these people know that?
The Chinese government?
No, the conspirators. Where do they get their information from?
I mean,
did you ask them? What are their resources?
No.
Ah,
we know that China.
Why would you do that? Of course, then they'd have to reveal the fact that they just made it up.
But we know that China has a one.
Do we know that? Do we really know?
They don't have a one-child policy anymore.
Well, then they don't have a one-child policy, do they? Because they realize that they've got a population crisis on their hands the other way that they didn't expect. They don't have enough people.
Well, the idea was
China
that the
does not
COVID
have enough
virus-
people.
But the idea was that the COVID virus was
That's a
was
tragedy.
killing. Yeah, it is a tragedy, absolute tragedy. But some people believe that the COVID virus was a way to kill off older generations who weren't doing anything productive.
These are people that don't like older people that made that up. They just made it up.
Yeah,
Conspirators make it up
they do.
because they have no facts. So they conspire. That's what that word means. Let me give you the example that I love the most, okay? You can take this to the bank and make a lot of money on it because you can confront any conspirator with this difference, okay? What is the difference between science and religion?
Empirical evidence
For?
I don't know,
No. For
Oh.
science or for religion?
for science.
Of course. Science is driven by observation, experimentation, and the collection of knowledge, which is testable knowledge, testable, period. It has no politics. It has no societal impact. It has no consciousness. It is what it is. Science is blind because it's just a collection of facts that's equally shared between people who want to know more and people who want to discourage other people from learning those facts. Why?
Why?
Because those facts disagree with religious tenets, which are all based on beliefs, not facts. So science is not a belief system, period. But belief systems are belief systems. So now, curiously, even though you weren't a good student, you must have been a fabulous thinker. Why? Because you've got your own podcast now and you're questioning things and you're opening up your world and you want a better education through this. I can sense that you're learning more than you're teaching. All right?
Yeah.
Okay. So take a guess, just a wild guess, as to how many religions there are.
Currently practiced?
You can look it up on your computer.
But I want to guess. Is it religions currently practiced?
I guess, because everything else is in the past. Can't change that. Today, existing in the world, how many religions do you think there really are? Recognized religions
I want to say
that
50,
have.
but there's probably
Come
way
on.
more.
Go to your computer. Don't trust me on this one because I'm a scientist.
Yeah. How many
I don't have a belief system.
are there? 4 minutes left, by the way. There are 10,000 distinct religions worldwide.
God bless you. You said 50. Now, how do they mean distinct religions?
I don't know. You'd
You'd have to go look that up.
Is Catholicism
how-
different than Christianity in this
No,
category?
Christianity is a generic term,
Okay.
and Catholicism is a form of Christianity.
Yeah. Okay.
But that still counts as one.
Okay, it's still one.
Right?
Yeah, that's a
Yeah, so
10,000.
10,000.
Which one's right?
Oh, you've asked the wrong question. That is a bad question because everyone thinks they're right, don't they?
Mm.
And so people can create situations which allows them to be cruel and insensitive to everybody else's belief system except their own. And that's not a religion at that point. That is a belief system that gives them a social advantage by imposing their will on less able people. And we know lots of examples of that. But it's still a belief system because when you start to pry down into the origins of that belief, it all goes back to mythology, primitive behavior, the belief in the stars, lots of different things influence the outcome of a person's belief system. And before the age of reasoning, which occurred back in, let's say, the 1300s, when the Renaissance occurred, people were not allowed to question the validity of their religion, so they went along with it. And a lot of religions were cruel. Like, for instance, the Incas. I'm not going to give you a lecture on religion, trust me, because
they were pretty.
That's my least favorite. The Incas were the cruelest of all the cruel people, except the Aztecs, to their own people, to get them to do what their religion says to do. And that is to allow me to eat you because there's not enough food.
Yeah.
When the Spanish came,
they were also cruel. The Spanish were the worst.
Whoa, but there was a big difference, an enormous difference. Why do you think the Spanish won? How could the Spanish possibly beat 20 million Peruvian Chileans with 200 horses and a troop of about 1,500 troops? How could those troops actually win? That's a great question, isn't it?
Didn't they take the king?
Oh, they took the princess. They took a room full of gold, and then they took the princess, and they said, we'll sacrifice this princess unless you fill this room with gold. In a week, they filled the room with gold, and then they killed the princess in front of about 100,000 people that had gathered to see the release of the princess. So why didn't they just overwhelm the Spanish at that point and say, goodbye, we don't need you? Because the alternative was worse. I learned this from an anthropologist that I actually listened to while I was on vacation in Peru. So when the people of Peru understood what Catholicism was, the big difference between their religion and Catholicism was, when you die and you've lived a good life, you go somewhere else. You get to go to heaven. They didn't even have that concept. There was no such thing as heaven. So when they died, there was nothing to live for. Their whole life was spent doing someone else's bidding and then dying for nothing. When the Spanish came along and said, oh, no, 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 no. That is not how religion works at all. Here's the way it works. We had a savior that came down and said, you are all bad people, but I will sacrifice my life for you, and you will then have a way of getting rid of your badness, and then you can become a good person. And when you die, you get to share heaven with me. Now, that's one slight exaggeration of one of 10,000 different versions of what happens to you after you die. Now, does science have an opinion as to what happens to you after you die? You're goddamn right they do. Oh, yes, they do. Your body decomposes, all of your elements get redistributed in nature, and you become resurrected as other life forms. That's your heaven. Now, your consciousness will not know what that is, but your atoms will thank you for recycling. And 10,000 religions are all based on mythology and our imagination as to what happens after we die. Every one of them. There isn't an exception. People used to think that because the wheat crop didn't come in so good this year, somebody fucked it up. And we were going to find out who that was. And when we do, we're going to kill them. So on the island of Crete, they said, you want to be king for a year? Oh, you can have anything you want. You can have 77 virgins. You can have all the wines you want to eat. But let me just tell you something. There's a little catch to this one. At the end of that year, we're going to heap all of our sins on your shoulders. We can do what we want too. And at the end of that year, you, the king, will be killed as a sacrifice to the gods. It tells the gods, we are sorry for what we did, and we express it by killing this person. That makes a lot of sense to me. And there's a wonderful book written called The King Must Die. And it's all about that religion, which doesn't exist anymore because people kind of figured it out. It's kind of stupid. So if you look at all the Christianity religions. All right. First of all, it was a rebellious religion, right? They were rebelling against the Romans, and they were being killed and slaughtered like crazy. The Romans kind of didn't believe in their mythologies anymore. Mercury was, come on, Mercury, that's Venus, give me a break here. They really had given up on their gods. And so people needed, the common people, especially the ones that were captured, they needed to believe in something that gave them hope and promise of a better life. And somehow this person who calls himself Jesus of Nazareth appeared and began to philosophize about the importance of life and the importance of being kind and the importance of generosity and the importance of being true to yourself. These are tenets that anybody could have done, all right? But one person decided to do it because he saw that there was a need for it. And it turned into a religion, and I don't think he ever intended that to happen at all. He just wanted to tell the people that being a human being and living a good life is enough. And yet the Romans, of course, thought this was subversive. And so that's how Catholicism started. But remember where it started to split. Luther, Henry VIII, all these people, because the religion started to set up all these blockades. You can't marry because you have property, and we need the property because we need to be rich. We need to be rich so that we can expand. Everybody needs to have more children. They begin to invent things that worked against normal culture. And so people began to invent other religions based on Catholicism, but eliminating the part that they found objectionable. Those are just whimsical things that people wake up one morning and say, I had a dream about. That's common. If you've got a strong personality, you can turn that into a religion. If you've got an even stronger personality, you can conquer your country with Hitlerism and Stalinism and Putinism and Genghis Khonism and all these other isms and Caesarisms and Kingisms. All these isms are based on power and control. That's my lecture for the night. I don't believe in science. I practice science. And I go wherever the science says to go, that's where you got to go.
I need to go for 1 second, and I will be right back, and then we will wrap this up. One
you
second.
Have no Australian accent.
Nope. Sorry about that, mate. I had to go and do my 5-show workout. And I had to get my Australian accent going again.
I like it.
American accent is just a ploy.
Are you an actress by profession?
No, I just
because you do a great American accent.
I've lived in Australia.
Nicole Kidman does a great American accent.
Nicole Kidman
she's Australian.
does.
She's fantastic.
Yeah, she gets paid the big bucks for a reason.
Okay, now here's the deal. We covered a lot of topics tonight, but we concentrate on gain of function, which is a trivial issue, by the way, as you've since learned. Because try as we might to replicate nature's progress, we have failed miserably in coming close to that. And the AIDS virus is the best example. We've worked on that thing for 30 years without success. 30 years, and we've had all the equipment you can possibly imagine. We've spent $1 trillion trying to figure it out, and we still haven't done it. We have 3 medicines that we give now, which completely inhibits the replication of the virus, but it doesn't get rid of it. It stays in the bone marrow, in those little cells where the virus does not replicate, and therefore the drug does not affect their ability to reproduce. And as long as that's the case, there will always be AIDS until the last person with AIDS dies. And that's not going to happen because people are continually catching it. They think triple vaccine. triple drug allows them to have unprotected sex. And that's why this virus continues to survive.
If you have AIDS and you're a listener to this podcast, stop having unprotected sex, please. Okay, go on.
Thank you. No, I'm done. I'm done. I'm off my soapbox. I have to go eat.
Next, perhaps if you do want to do a podcast next time.
Serene, it'll be a long time before I do a 2-hour show with you. But I would do a 1-hour. I would do a 1-hour show.
I would do a 1-hour show.
And it has to be a selected topic that has relevance to what I'm interested in this time. Because this was all about you. It was all about, can I answer my friends' questions about gain-of-function research that doesn't panic them into thinking that the scientists of the world are trying to poison us? Because that's a conspiracy.
What's
I don't want to be poisoned by Dr. Mengele. And it's never happened before. Why should it happen now?
What is the next topic?
Even if we wanted to do it, we couldn't do it.
No, I don't think so.
That's the problem.
I think you gave it pretty good.
There are people out there that want to do it, and we don't know who they are. If you say I'm against gain-of-function research, do you think for a minute that would stop those people from doing it?
There are
you kidding? Spur them on and let's do it more.
Any population is going to have psychopaths and extremists who want to kill the world. I mean, you could say that gain-of-function research might be stopping malicious actors from perhaps being able to infect people as much. Anyway.
They do it all the time for germ warfare. They have bombs they have lots of infectious agents like anthrax and things like this in them, and they explode them in other people's worlds. But sometimes the wind blows in the wrong direction and the goddamn microbes come right back to the people that spread them and they kill them instead. That's what happened in Germany during the gas attacks.
Chlorine gas, yeah.
It didn't always work in favor of the enemy, you know.
No.
Sometimes the Germans wiped out themselves with mustard gas because the wind changed.
Yeah.
All right, so that's stupidity personified. That's the foible of mankind, or humankind, I should say. So I have to go.
Is the next podcast on vertical gardening?
No, we're not doing those anymore.
You
can go look them all up. You can go look them all up, though. The next podcast is Friday, which is tomorrow.
No. When we do a 1-hour podcast next time.
When we do ours.
What's it on? What's the topic?
I'll let you know. Let you know.
All right.
And it won't be for at least a month.
Fine. I'm perfectly fine with that. Hey,
so
Dickson, thank you.
Wait a minute. I have another question. Question for you.
A question, yeah.
Since you're new at this, do you know how many people are listening to your podcast?
Very few.
Why do you think that is?
I would love to know.
Yeah. So how do you get your podcast out there?
Right now I don't have a lot of channels. I'm doing it on Reddit, some of them on Reddit, and not a lot else. You need
help.
Yeah.
You need some technical help. So you need a partner that's already succeeded at this. And maybe you should need to join their group. And become a subsidiary.
Do you have any recommendations?
None. I'm a scientist.
I've looked.
I know our own podcast. Let me just give you some history. Our own podcast started with This Week in Virology. That's the first time I ever did anything like that, right? It started in like 2006. When we first started, you go back to episode number 1. We have archives that'll take you right back to the very first one. And you can listen to me with a good voice. We had a counter, and the counter told you how many people downloaded that version of your podcast, right? We were in the hundreds for many weeks. Nobody knew what the hell we were doing, and we didn't either. But we decided to advertise on other people's podcasts. So we got our podcast listed, and we gradually built up the numbers. Over the last year, take a guess how many people downloaded our episodes.
In total?
Per year.
Per year.
6 million.
Wow. Oh my God. You guys are
Yeah.
Wow.
That's big time. That's big time.
Great.
This Week in Parasitism is in the hundreds of thousands.
Oh.
We don't go on as regularly, but that's what happens because the topic spread. I was going to use the word viral, but in that case, it didn't. It spread parasitically. We have clinical cases. We give clinical cases each week for the This Week in Parasitism podcast, and then we ask our listeners to write in their answers, and then we give away a free book. So you have to have gimmicks, you have to have hooks that get people involved. I think a great way to present your podcast today is to title it something.
Yeah, the title would probably be good.
Now I'm going to give you a title for this one.
Okay.
What's the difference between religion and science? And they're not going to find out the answer until they listen to the entire podcast.
Ooh, I like that. Okay.
So put it out there as a teaser.
Let's do it.
And it'll completely blow the mind. Well, some people don't care. I mean, there are a lot of people that don't care, actually. They want to hold on to their own ideas, and they don't give a goddamn what anybody else thinks, because it's their idea, and it's my idea, and it's right, and you're wrong. Well, scientists think that way too, by the way. When they come up with an hypothesis for something, they know theirs is the most correct, so they badmouth everybody else's.
Yeah.
But you know what? The other person does the experiments anyway. And they say, oh, guess what we found? We found that you were wrong and we were right. And we love saying that to other scientists because it justifies and it validates our existence. And so when a religion conquers another religion, it validates the fact that God's will allowed that to happen. And the Arabs have a wonderful tag end to this story. And it's an expression that you've heard. It's called Inshallah. Inshallah. One of my favorite movies, if I had to list 10 movies that I love, one of them is called Lawrence of Arabia. It's Peter O'Toole. And there's a scene in that movie where Omar Sharif, no, I think it's not Omar Sharif. I think it's Anthony Quinn, says to Peter O'Toole, after a little boy was saved because he fell off his camel after they got the watering hole and he was going to die of dehydration. Peter O'Toole went back and picked him up and brought him back. Later on, somebody had to shoot him. And Anthony Quinn came over to Peter O'Toole. Peter O'Toole had to shoot the boy. And Anthony Quinn said, it was God's will. And you can excuse a lot in this world by saying it's God's will, but you can't excuse anything by saying that's a fact. So if people choose to behave that way, that's fine. But it doesn't mean anything to me. I want to know where you heard that. I want to know what was done to create that piece of knowledge. And if you can show me the path, the paper trail that leads back to the origins of that thought, I might pay attention. But if you just come up with an idea, like I had a dream last night that I became superpower and I can fly off a building, watch, I have a cape. You know how many little kids have died doing that in the world? They see a cartoon, they see a guy put on a cape and they fly off a building. And the next thing you know, they're trying the same thing. And of course, they end up killing themselves. That's the difference between belief and science. The scientist observes the action and then never puts that cape on. And there are lots of stupid people out there that will drink the Kool-Aid. Remember the colony in
Jonestown.
Who could forget? Who the hell would ever participate in something like that? Are you kidding? There's lots of gullible people out there.
But almost everyone did. Almost everyone killed themselves. They went against their own instincts, their own evolution.
They did. They did. And on that note, it was a pleasure.
It was.
I have to leave, otherwise I'll keep talking.
This is a wonderful podcast. Thank you so much. Have a good night.